students, and welcome to this mini lecture on the inheritance basis or the genetic inheritances of disease. The learning outcome that I want you to take away for today, so the learning outcome is to be able to explain the inheritance patterns of different diseases. Okay, so what do I mean by this? Well, inheritance can be either recessive or dominant, and I'm sure you've heard these terms. And in terms of the genes that we're referring to, where are they located, on what chromosomes? The chromosomes can be either autosomes or sex chromosomes. So the inheritance patterns that we're going to go through today are autosomal recessive or dominant, or sex chromosomes recessive or dominant. Now, because most of the sex chromosome is going to be affecting the X chromosome, because the Y in the male doesn't really do a great deal in terms of the inheritance patterns, most of what these chromosomes will be referred to as X recessive or X dominant. All right. So let's get a start, let's get a context. So in terms of inheritance, we're referring to genes, which we're referring to DNA. Now we know in our cells, 30 trillion cells, all our cells except red blood cells have DNA. Now, if you were to put this DNA stretched out in a linear fashion, so if I was to stretch out all your DNA in a cell, a big long line, it would be approximately 2 meters in length. Now that's a lot of DNA to try and to get into a cell that's only like, let's say, 5 microns, which is like thousandths of a meter. So we've got to somehow jam it into the nucleus. This is the nucleus here. How do we get it in there? Well, we do it through folding the DNA into chromosomes. So within the DNA, we have chromosomes, which are segments of DNA that are squashed together, and we call them chromosomes. Now, if we were to do a karyotype, which is making sense of all your chromosomes, what we would see is something like this. This is a karyotype below in this picture. So what I'm seeing here are the chromosomes. So this is chromosome 1, this is 2, this is 3, this is 4, continuing, all the way to, let's say, we'll stop at 22. Now, these chromosomes 1 to 22, okay, so write this down, 1 to 22, are known as your autosomes. So let's write this down. Number 1 through to 22 are your autosomes. Okay, so we're left with these at the bottom. Now, please see that it says or, which means you've got two options for the chromosomes at this end, which are the sex chromosomes. So here you can see XX. So these would be your sex chromosomes. Sex chromosomes. Okay, so XX would be a genetic female or a biological sex female. And XY would be biological sex or genetic sex male. All right, so that's your sex chromosome. And as you can see, you've only got one paired here, whereas autosomes, you've got 22 pairs. So what do I mean by pairs? Well, simply one of these, let's say that one is from mum and that one is from dad. So you've got a repeat on every chromosome. One you get from mum, one you get from dad. Okay? So that's the chromosomes. Now, we're going to get to the inheritance now. So let me say, today's example, at least for the autosomal, either recessive or dominant, we're going to focus on chromosome 1. Now, I'm just going to make something up here. It's not genetically accurate, okay? So I'm just going to make it up, but the principle behind it is true. So on chromosome 1, we've said one of these chromosome pairs is mum and one's from dad. And let's say we'll focus just on this segment here. So the smallest segment of the DNA within that chromosome that codes for something is what we call a gene. So it's a line of nucleotides, I'll go through that in a second, which code for which makes a gene, which then essentially makes a protein. Okay? Now, just that little part of that chromosome 1 is the gene that we're referring to. So let's say we were to zoom out now, and what we can see is a segment of DNA. So this part here is a gene. Okay? So all of this content of base pairs, and what I mean by base pairs are these colourful things. See these colourful things? That is what gives the alphabet of DNA. And this is essentially these. So it's like G, A, C, T in a whole different array of orders. But the smallest segment of DNA that codes for a protein is a gene. So for today, I'm just going to say this segment here is a gene, and we'll call it gene A. Okay, gene A, and it's going to make something which is important for function. So I'm going to jump across to the next screen, and we'll start with the autosomes, and we'll look at recessive and then dominant, and then we'll go to sex chromosome recessive dominant. All right. So we're focusing on chromosome 1 and specifically gene A. Okay. Now, if we were to look at what the gene could make, I'll change color. I'm going to show you what the actual nucleotides are. So let's say Dad gives you G, A, C, T, A, G, A. Let's just say that's one gene on that one chromosome. So let's say here's Dad, and over here is Mum, and we're just focusing on that one there. So that's Dad's one, and then Mum's one is G, A, C, T, A, G, A. Right. What you notice is they're exactly the same. So what this term means, if the gene's exactly the same, it's called homozygous, which means identical. Now, if you were to express this gene, it would make a protein. Okay, and same with this one, it would make a protein. Let's say the protein it makes is a special protein on a red blood cell. So here's a red blood cell, and here's a actually, I'll just say it combined. I'll put it in the middle. So let's say these proteins together will make here's a red blood cell, and the protein it makes helps to formulate its outer covering, which makes it a nice, robust, kind of concave shaped red blood cell, which is really important for its function because we know red blood cells need to fit down small capillaries. So these genes are working in a good working order. Okay, so the red blood cell is shaped as it should and it's working fine. But let's just say there was a slight alteration in the genes for Dad. So instead of A there, we've now got, I'll put another T. So what this has done is it's caused a change in the code for this gene, and it's going to change the way that the protein is made. Now, before I get ahead of myself, now because this gene here is different to Mum's, we're now what we call heterozygous. So we are different now. Okay? Now, let's say this change has caused a loss of function in the protein. So let's say the protein doesn't work now. Okay, but luckily Mum's gene is functional, so it's good. And what that means is it overruns or over controls or over dominates this loss of function from Dad, which means the red blood cell is still good. So let's just put this out here. So we're going to put, for the gene, it's called gene A. Let's call the functional gene A little a, and the dysfunctional one the big A. So because this is coming from Dad, we'll call Dad up here, and we'll call Mum down here. So this is the chances of offspring from Mum and Dad. So Dad has a small a and a big A, whereas Mum has a small a and a small a. Okay, so what could happen here is that of the offspring, you could have small a, small a, small a, big A, small a, and small a, big A. So what this means for the offspring is, for this individual, completely fine, no problem at all. Same with this one. Whereas these two individuals have one dysfunctional gene, gene A, and one dysfunctional gene A. So what this is saying is 50% of the offspring would be completely normal and 50% would have a dysfunctional. And what this means is because this is overpowered by this one, these two would be recessive. Okay, recessive. because this smaller a dominates the bigger a, like we saw over here. So this is mum was a small a and dad was a big A. Okay, so that means these are recessive, which means they don't have it. They carry it, but they don't display it. Okay, hopefully that part makes sense. What if we saw the same thing across here with mum? So we saw that she had another T here, which made it a loss of function here. So mum has a loss of function, dad has a loss of function, which means what for the red blood cell? Okay, well, let's put it in the Punnett square now. So coming back to the Punnett square, so the options we could have is one normal gene, one abnormal, and for mum, one normal, one abnormal. So here we've got still a normal one, but here we've got a recessive, here we've got a recessive, and now we've got a double A. So what does this one mean? Well, if both of them are a loss of function of a gene, which is these guys here, what you'll see is now the protein doesn't work. And let's say the red blood cell now looks like this, which is what we call a sickle cell red blood cell. And this would be an example of a recessive autosomal inherited disease because it's on chromosome 1, which is an autosome. It's recessive, which means you've got to have both of the defective genes to get it phenotypically. What I mean by this, phenotype means appearance of, whereas genotype is what your gene is. So for here, this is the genotype of small a, which is normal genes for both. The genotype for this one is normal gene, abnormal gene, but because this overweighs this one, your phenotype would be still normal. Here, the genotype is small a, big A. Again, the phenotype would be normal because the small a dominates. But here we've got only 2 defective genes, which means the phenotype is you've got the disease in this recessive form, which because the protein is a loss of function from a defective gene and a loss of function from a defective gene, you've got no good protein anymore for the red blood cell, which makes the red blood cell sickle-shaped. Okay, so hopefully you can see that being the recessive characterization of an autosomal recessive disease. Another example of a autosomal recessive would be cystic fibrosis, which is an issue with kind of chloride channels, and that can affect lungs and pancreas and so forth. Let's have a look at a autosomal dominant. So I'll get rid of all of this. Now, let's say for this gene now, I'll keep the genes the same, but let's say the change that we've seen, the change which is change into a T, okay, we change it to T, but this time the change in the T is causing a gain of function, not a loss of function. So it actually causes the protein that it has now made or code for to become a different form that is now activating the protein, not turning it off. And so in this case, and we'll go back to the mum here and just put it back to normal, put it back to an A. Okay, so it's only dad that has the defective gene. But because this particular gene is now a gain of function, and the mum is normal, the gain of function of this particular gene into the protein makes it a dominating gene into a protein, which makes the actual outcome, let's say the red blood cell, to become defective, which let's say is an odd-shaped red blood cell. So the take-home point here is for a genetic change that causes a gain of function, not a loss of function, a gain of function, you only need one of them to over-dominate the other. And that's where you get the dominant recessive. So let's go back to our Punnett square. So again, we've got dad here with an AA and mum with an AA. So what we've got as an outcome here is AA, or big A, should I say, small a, and small a, big A. So the outcome is genotypically normal, 2 small a's, genotypically normal. So with these 2 normals, phenotypically, it's normal. So 50% of the offspring will have none of the defective genes and they will be phenotypically normal. But over here, we've got a genotype of one good gene, one bad gene, and same here. So it's 50% abnormal, but because it's dominant, big A, and it's because it's a gain of function dominant, that means that overweighs this one. So that means 50% or one in two of the children will have the defect. And that's going to be the autosomal dominant. And so an example of an autosomal dominant disease is Huntington. So what that basically means is if the gene from one parent is defective, even if the other is fine, the fact that it's bad on the dominant gene, because it's a gain of function gene, it means it overpowers the other, regardless if the other's normal. And that's exactly the opposite in recessive, because remember, recessive is a loss of function, and the good gene can actually overcome the loss of function in the bad gene. So that's the difference between the recessive and dominant in the autosomals. So now let's go to the sex chromosomes. So the sex, as I said, we're not going to focus on Y because Y doesn't really code for a great number. So what we're going to focus is on the sex chromosome. Okay. So here are the X-linked. Okay. And again, I'll just go straight down to the Punnett square. So here's Dad. So Dad, we know, has to have a Y. So we've got an X and we've got a Y, and Mum is XX. So let's say the dysfunction of the X chromosome is on Mum this time. So Mum has a defective X chromosome that's depicted by that black circle. And let's say this defective gene is for clotting. So the protein we're looking at is for clotting blood. Now, so far, we're going to call it a recessive. So this is a X-linked recessive condition. So what that means is if it needs to be carried on two of the genes for it to be seen, okay, opposed to a dominant. So the outcome here would be, let's write it out, we've got XX, we've got XY, we've got XX, and we've got XY. So the outcome, as you would expect from Mum and Dad, is 50% of the children are going to be male sex and 50% are going to be female sex. But we're also looking at where you see these black parts on the X chromosome, which is depicting the defective gene. And so what's the outcome here? Well, we've got one bad X, one good X. Here we've got one bad X, but we've got no other X. So what that means for this particular offspring is it's going to be genotypically X defective, but we've got no other X, so it's going to have no other option. So this outcome is this male will have it. Now, because males don't have any other option, there's no male recessiveness here. It's just a dominant, straight away dominant, because there's no other option. So X-linked in males is always going to be dominant, okay? This male down here is okay, so unaffected, both genotype, phenotype. Over here in the females, this is genotypically one X is defective, the other X is fine. So genotypically, it's 50-50, but phenotypically, the other one outpowers it. So that means that she's just a carrier. This one is a carrier. Oh, I rubbed it out. This one is a carrier, and this one is unaffected. So the outcome here would be 25% is going to be affected, both geno and pheno. Okay. 50% are normal, both geno and phenotype, and 25% are carriers, which is this one, is a carrier. So these two are normal, this one is affected, but both genotypically and phenotypically, and this one here is the carrier. Okay. Now, if it was dominant, then we would see for the female. So let's do it again. X, 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 Y. Let's say it was a dominant gene just on one. So we're doing the same essentially. So X, 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 Y, X, 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 Y. Now we'll put, so there's the one, So if it was a sex-linked dominant, so this one here is going to be the same. So this would be a genotypically and phenotypically affected male. This one here, because it's a dominant effect, this one here means it's now affected female, both genotypically and phenotypically, and these two are still unaffected. So 50% would be unaffected, 50% would be phenotypically and genotypically affected. So there you have it. Hopefully now, when we go back here, you can see the difference between dominant and recessive. That basically comes down to the loss of function for the recessive, gain of function for the dominant, and you can see how they operate different on the autosomes. Examples being sickle cell anemia and cystic fibrosis, whilst the sex chromosomes could be examples in a recessive would be hemophilia, but for the male, it automatically becomes dominant because it's X-linked. Remember, the males have no other option. And another recessive X-linked would be muscular dystrophy. So there you have it. Thank you, guys.